Hello, this is Tim Sandle. I'm really sorry I can't be with you at the conference. I hope you're all having a great time. So I'm here virtually and I'm going to be presenting on Cleanroom microorganisms and the importance of identification and for screening and characterizing and trending microorganisms in general. So the presentation is going to be looking at contamination sources, contamination control. We'll touch briefly on the human microbiome and the microbial ecology of human skin. And we'll have a look at the types of microorganisms that are found in clean rooms. And we'll kind of wrap up by saying why microbial profiling, screening for clean room microbiota matters. So let's begin with looking at some contamination sources, because the different sources of contamination coincide with some of the types of microorganisms that we might expect to find. So we have a contamination source with air, particularly as a vector for moving microbial carrying particles in air streams. We also have risks from air moving from less clean to clean areas. We also have equipment transfer, particularly moving items through the cleanroom cascade. We have surfaces where contamination can settle through deposition or from touch contact, touching a contaminated surface, going on and then touching a supposedly clean surface. And then we also have water and water sources. And water is both a growth source for many types of microorganisms, and it's also a vector of contamination through leaks and aerosol transmission. But most importantly, the fundamental source of contamination within clean rooms is from people. Now, understanding these sources of contamination is essential for putting together the contamination control strategy. And as you know, and as you surely would have heard throughout the conference, that the contamination control strategy is a fundamental of the Annex 1 revision. And by control strategy, one thing we're seeking to do is to show that every element is connected, everything is interlinked. And understanding the manufacturing control strategy, the quality control strategy, setting quality attributes, the design of clean rooms, the risks of cross-contamination, product segregation, and so on, is all connected to minimizing contamination. And one of the key steps in that is with understanding clean room microorganisms. And on the slide there, we've got the kind of representation of how things are interconnected. Now then, I mentioned people and the criticality of people. So we are a fundamental source of contamination, probably representing somewhere between 70 and 80% of the contamination typically found in clean rooms. And that's because across the surface of our body we're harbouring at any one time at least 30 million bacteria. And we are shedding millions of skin cells per day. And the work that was undertaken in the University of Glasgow by Bill White has shown that around 10% of microorganisms, 10% of skin particles will be carrying microorganisms, and around 4 microorganisms per piece of skin matter, with the average piece of skin matter being around 4 microns. And in addition, if we're not gowned correctly, we're not wearing masks appropriately, then we'll also be releasing particles from our mouths and nose as minute liquid droplets, referred to as aerosols. Now, there are other parts of the body that harbour greater numbers of microorganisms, as we can see on this slide here. So this is information that's been taken from the Human Microbiome Project, and you can see the proportion that's in the gut. But the skin microbiome is still substantial and thus presents a substantial risk within the cleanroom environment. Now, we're not going to be able to recover everything from the cleanroom, and most environmental monitoring is going to be biased towards those organisms that are predominant on the skin. And the reason for that is to do with variations with our monitoring methods. So most environmental monitoring methods are only going to capture around 50% of what could potentially grow. And in terms of what could potentially grow, that's heavily influenced by whether the organism is culturable. So it could be in a state of non-culturability, which could be something that is transient, like a stressor has rendered an organism that's normally culturable no longer culturable, or it could be simply that the organism there's yet to be a culture medium against which this organism can grow. So the types of agars are influential, but so is the location of the monitoring sample, and picking the right location matters, and also our sample sizes are going to be relatively small. There's other factors about whether we've got the presence of an antimicrobial, such as disinfectant residues, so we need to have appropriate neutralizers in particularly surface samples and plates used for finger plates. And we're also going to get variations in recovery depending upon the incubation temperatures, if we're doing dual incubation, the order of incubation, the incubation time, and then also with the identification method, because a number of identification methods are relatively limited. Many have databases that are biased towards the clinical. So we're going to have these considerable variables. Now, another key factor for what we find is going to relate to which parts of the human body are most likely to release microorganisms. Now, to a degree, this comes down to the efficacy of gowning, but it still stands that the human skin, this very large organ, this 1.8 square meters on a typical adult, is a series of different ecological niches. So the temperature range across skin is from 25 to 37 degrees Celsius, with the warmest areas under the arms and the coolest areas at the ends of our fingers. The pH of the skin also varies, ranging from the acidic up to just around neutral. So the forehead has a pH of around 4.8. Under our arms, then we're close to neutral. And we also have variations in moisture as well. So again, armpits, but also between our toes and other areas, we have high moisture levels and therefore high population densities. But other areas, say on our forearms, are relatively dry and they have slightly lower population densities. And we're also going to get different rates of skin shedding as well on different regions of the body. So the importance of gowning is a lot of it is torso dependent, arm dependent, because these are the areas that shed more readily. Now, the studies into the human microbiome of human skin have shown that there are up to 1,000 different species on typical people, and that's based across a number of geographical habitats. But these can be then divided into around 19 phyla, or groupings of similar organisms, genetically similar. However, there's 4 phyla that make up the vast majority, and these are the actinobacteria. So these are gram-positive organisms like Micrococcus, Corynebacterium, Propionibacterium, Cutibacterium. Then there are firmicutes, who are the organisms like Clostridia and Bacillus, proteobacteria, which is some gram-negative bacteria, and Bacteroides as well, which is also a group of gram-negatives, which are different environments. As I mentioned before, these different ecological niches. So forehead and scalp, predominantly sebaceous glands, and they're dominated by Propionibacterium, Cutibacterium, some Corynebacterium. So we've got some interesting thoughts there about anaerobes as well, and the risk they might present. Between the toe webs, we've got this great diversity, and here we find some gram-negatives that have an association with the human body, particularly actinobacteria. And also we're going to find a lot of fungi. So the greatest concentration of fungi on the human body is on the feet. Arms and legs, torso, relatively dry. This is where the main cleanroom recovered organisms tend to predominate, the staphylococci, the micrococci, slightly lower population densities, but key organisms for cleanrooms. Sole of the foot, there's a lot of staphylococci. Main body, then again this Corynebacterium, some fungi, some Acinetobacter as well. So we've got different body regions, they're going to be released at different rates, and we've got some interesting thoughts there about gowns and control that we need to have a think about. Now, why do microorganisms survive, and conversely, why do some microorganisms not survive in cleanrooms? So survival is dependent upon species and time, but there's a range of influencing factors. So the temperature, the availability of water, the concentration of organic compounds and of hydrogen ions, the level of inorganic compounds, the redox potential, the pressure, light intensity as well. So these are all factors that can influence survival, might trigger why a particular organism, if it's capable, goes into the spore state. And also we have some variations with different types of clean rooms as well. So we have factors in terms of clean room grade, the temperature and humidity of the clean room, the gowning requirements to go into those clean rooms as well. So you know, there's a lot of factors that can vary what we might recover. And also mentioned about microbial identification as well. So with microbial identification, there are going to be variations in the method. So that's very much dependent upon the database, whether it has a clinical bias or not. Even very popular technologies like MALDI-TOF are still limited to a degree in what they can recover, and sometimes they throw up very strange organisms, which is why you would need a microbiologist to interpret those findings. And the issues of culturability we've also mentioned, and there's also to a degree going to be variations in seedability as well. So there's going to be factors that trigger variation, but that shouldn't detract from our ability to review, detect and trend. Now, in terms of the typical organisms that you may well find, then I did a review, and this is part of the outcome of that review. So this is grade A and B areas, nearly 7,000 organisms profiled, and in these areas where there's no water sources, it's predominantly personnel undertaking the running of machines to carry out aseptic processing. Then 80%, over 80% of the organisms are gram-positive cocci. Then there's a proportion of gram-positive organisms and very low levels of gram-negative organisms and fungi. So ideally we don't want to see many organisms in these kind of areas, but if we do, then it's understandably those that have been released from the human body. In other particular species, then from my review of these 80% of these 7,000 isolates, then you get Micrococcus luteus, Micrococcus lyell, Staphylococcus species, Micrococcus species, Staphylococcus epidermidis, Staph capitis, Staph hominis, Staph hemolyticus were the predominant organisms found, with a low level of occasional spore formers, which can either relate to what we're carrying or what's been transferred, so it could be breakdowns with transfer disinfection processes. In C and D clean rooms, then from around 2,500 isolates, there's much greater diversity, and one of the factors that affects that diversity is whether there's water sources, and secondly, you've got lower levels of gowning, and you've got perhaps more variability with transfer of items. But really what you're hoping to see is, as you transfer items through the different clean room grades, you're seeing a reduction in count, and you're seeing a reduction particularly in spore forming microorganisms. So what could we draw from these types of reviews? Well, we see an association between the microorganisms commonly found in clean rooms and those that are transient or long-term residents on human skin. Relatively low levels of bacillus, but we still do see some bacillus, so it could be us, as I said, or it could be transfer of items, dust, and so on. We will occasionally, very, very rarely, get nasty microorganisms that might be resident from within the human body, and that's going to come down to gowning, mask control, and people's personal hygiene. And where there is a water source, then we will occasionally get water organisms that will be recovered. Another interesting thing about profiling microbial contamination from clean rooms is that there's often little variation over time. When variation occurs, it's normally because something fundamental has gone wrong. So it could be a change of cleaning techniques, a new shift starts, problem with air handling systems, HVAC failures, and so on. Another interesting factor is the types of samples. So personnel samples, exit suit plates, finger dabs, and so on, then predominantly, overwhelmingly gram-positive cocci. Air samples, again, mostly gram-positive cocci, particularly active air samples that are going to be picking up people shedding. Surfaces, relatively high in gram-positive cocci, but here there can often be a reasonably higher proportion of gram-positive rods and particularly occasional spore formers. And where gram-negative rods occur, unless they are things like Acinetobacter that might have a personnel connection, so you might find Acinetobacter in changing rooms along with Paracoccus yaei, then most gram-negative rods do have a water connection. Another interesting feature of the cleanroom microbiota is that the majority are mesophilic organisms. There's a few that are facultatively aerobic, so we'll get some Cutibacterium, for example. And we tend to find very few organisms that you would regard as extrema tolerant. In terms of fungi, where fungi are recovered, then Aspergillus, Penicillium are the highest, sometimes Cladosporium. Sometimes in changing rooms we may well recover Trichophyton, which is the organism associated with athlete's foot. So it's important to trend and characterise microorganisms and build up an indicator of what's normal and what isn't normal. So we're looking for changes from the norm. Changes from the norm may signal cleaning or disinfection concerns, gowning controls, introduction of new shifts, maintenance work, all these kind of factors, all these things we can build in and overlay onto trend charts. We also need to carry out extensive identification if we were, say, to have something like a sterility test failure. And here we need to go down to the genotypic level. And also we can screen microorganisms to help us with investigations and to look into out of limits results. So if we start recovering high levels of micrococci and staphylococci, then we're automatically drawn far more towards what have people been doing. If we're getting bacterial spores and in pockets or isolation, then we want to look about how we're getting things in. Has cardboard ended up into our clean room? Have we got a problem with our air handling system? Gram-negative organisms might drive us towards water leaks, drain suckback, and so on. So there's lots of things we can draw in, and this is where an experienced microbiologist is really important. And we also need to understand what we're going to identify and when, because we're going to be generating hundreds, thousands of samples, and we can't identify everything. Identifying things at alert and action level goes without saying, but we still need to identify a reasonable number of other organisms so we can build up a profile so that we can understand these drifts from what is normal or not normal. Okay, so thank you for attention. It's a bit of a whirlwind. We've had a lot to cover in this session, but we've outlined the human microbiome. We've looked at the what we theoretically might find based on the genetic profile of the human body and what cleanroom studies suggest we will find. We've looked at the diversity of different organisms, and we've looked at what these kind of things will be signalling. People, gowning, how we do things, disinfection regimes, air handling systems, and putting it all together as a fundamental to support the contamination control strategy. So, thank you very much. I'm Tim Sandle. Sorry I've not been able to be with you, and I hope you enjoy the rest of the conference. Cheerio.